So part of cell theory says that all cells come from pre-existing cells. So in this chapter, we're going to look at how existing cells give rise to new cells through a process called cell reproduction. This process we see in multicellular organisms as a way of growing the organism, maintaining, repairing tissues within the organism. In single-celled organisms, this is a process by which the actual organism reproduces itself. In bacterial cells, we're going to see this process as binary fission. So remember, in prokaryotic cells, the genome, the genes necessary for the cell to be itself, is in a single circular piece of DNA. It's not in a nucleus because it's a prokaryotic cell. It's in a region called the nucleoid region. And this DNA molecule must reproduce itself in order to reproduce two new cells. There may be some other pieces of DNA called plasmids. These are extra bits of information, not necessary for normal growth of the bacteria, but adds extra traits to the bacteria. So the process by which bacterial cells divide is called binary fission, and it's a 5-step process, fairly simple, especially compared to eukaryotic cells. And we're going to go through these 5 steps in this lecture. So the first step, what has to happen is that the chromosome has to completely replicate. In order to have two new cells, they have to each have a copy of the instructions. And this replication begins at a point in the DNA called the origin of replication. And the DNA replication occurs in both directions from that point of origin. So it's a very rapid process. As the DNA completes its replication, the cell begins to elongate to create space for the two new pieces to separate from each other. And at the same time, a set of proteins called the FTSC proteins start to migrate toward the center part of the cell. It's going to be between the two pieces of DNA when they finish replication. At the point we see here, the DNA molecule has replicated. It's now separated into two ends of the cell. And these FTSC proteins begin to form a ring that then begins to pull the membrane inward between the two pieces of DNA, which we can accept as happening because the membrane is very flexible and so fluid. As the membrane continues to fold in, the FTSC proteins are going to start directing the development of the cell wall at a region called the septum, which is going to then further divide the two cells. Finally, in the last step, the wall has formed. There's two separate cells, each with their own copy of the bacterial chromosome, and that completes the separation of the cell from one cell into two. So through this process, called binary fission, the chromosome of the bacteria is copied, replicated, separated into two sides of the cell. The membrane folds in between the two pieces, a wall is formed, and we end up with two new bacterial cells.